Today, I am going to discuss with you an important topic as far as the NEET is concerned. The chapter is body fluids and circulation, and the topic given it here is blood grouping. There are two types of the blood groups which are discussed in NCERT. One is the ABO blood group system, another is the RH blood group system. RH blood group system, as the name suggests, it was discovered in rhesus monkeys by Carl Landsteiner. He was awarded with Nobel Prize. It is a type of antigen called RH antigen present in human blood erythrocytes. Based on the presence or absence of this RH antigen, the erythrocytes or RBCs, they are of two types. One is those RBCs or erythrocytes with RH antigen on their surface, another RBCs or erythrocytes without RH antigens on their surface. So this type of person, they are called as RH positive person, and this type of person, they are called as RH negative person. As far as the demographic data are concerned, 80% population of the world is RH positive and 20% of the population in the world is RH negative. We can say like this, at least in India. As far as the RH antibodies are concerned, there are no pre-formed antibodies in any RH group. What does it mean? It means that if a person is RH positive or RH negative, there are no preformed antibodies present in his blood plasma. Now, the blood group, RH blood group is also inherited in a Mendelian fashion. The genotype for having RH antigen on the RBC surface is capital D or capital D small d, means homozygous dominant or heterozygous condition. In these conditions, the blood group will be always RH positive. If the genotype is small d, in this case, the phenotype will be RH negative. So this genotype, capital D, small d will give RH positive phenotype. Small d genotype will give RH negative phenotype. Suppose we say that if in any case the blood of RH positive person get mixed with RH negative blood, in this case the antibodies will be formed in the person having RH negative blood. Now the antibodies will be formed. As I told, there are no preformed antibodies. Now the antibodies, RH antibodies will be formed in RH negative person. This is a life-threatening situation. It is a fatal condition where there will be a mass destruction as a result of antigen-antibody reaction. There will be mass destruction of the RBCs of host. When the blood of RH negative person get exposed with the blood of RH positive person, If in any case blood of Rh positive is transferred to blood of Rh negative person, antibodies will be formed leading to mass destruction. Of host RBCs. This is a fatal condition. This is a life-threatening condition. Fatal condition. A critical case has been studied in detail in a specific situation when a person having Rh antigen on its RBC get married to a woman having Rh negative blood group. In this case, what is going to happen? Let us see. If father is Rh positive and mother is Rh negative. In this case, the father genotype, suppose capital D, mother having genotype of small d, and the first child is definitely going to be Rh positive. It is due to having genotype of capital D, small d, heterozygous condition. But this child will be born without any defect. It is absolutely normal child without complication. This is a condition which is safe until the delivery of the first child. Now, at the time of delivery, suppose this is uterus, this is vagina, this is suppose placenta, and this is umbilical cord, and this is fetus. At the time of delivery, this is the second pregnancy. Complications starts. Now, this is the time of first delivery. This is the time of first delivery. That when this placenta got separated, the fetus, we have already discussed, the fetus is Rh positive, mother is Rh negative. At the time of separation of placenta, at the time of delivery, the some amount of blood, Rh positive blood some amount of RH positive blood get mixed with RH negative blood of mother. And here the problem starts arising at the time of first pregnancy. The second pregnancy complication that is going to happen in this case, that the mother started producing RH antibodies against the RH antigen which has entered in her body at the time of delivery of her first child. At the time of delivery of her first child, the fetus is RH positive, mother is RH negative. Some amount of RH positive blood of fetus, it is mixed with the RH negative blood of mother. The mother start producing antibodies. Mother starts producing antibodies. These are RH antibodies. What is going to happen in second pregnancy? RH antibodies, these are IgG type of antibodies. They are transplacental antibodies. They can cross the placenta. They can cross the placenta and they can reach to the fetus through umbilical cord and placenta. RH antibodies, they are of IgG type, means they are transplacental antibodies. This is a crucial point to be well understood over here, that during second pregnancy, RH antibodies which are made by mother, these are the questions which are frequently asked in NEET type of examination. RH antibodies, they are produced by mother. They are IgG type of antibodies. They are transplacental. They can cross the placenta and they can reach to the fetus. Reach to fetal circulation. They can reach to fetal circulation. Now the problem starts coming. What will happen? There will be antigen antibody reaction. Antigen antibody reaction. This reaction will occur in fetal body. This reaction will occur in fetal blood. There will be mass, you can say, destruction of the fetal RBCs. Mass destruction of fetal RBCs. This process is called as hemolysis. This process is called as hemolysis, destruction of RBCs. It leads to development of a disease called Erythroblastosis Foetalis. Erythroblastosis Foetalis. This is the disease. Erythroblastosis Foetalis is the name of the disease, or we can say it is hemolytic anemia. It is due to It is due to RH incompatibility. Incompatibility means when the RH antigens, the collides with the RH antibodies, this leads to mass destruction of the fetal RBCs. The disease in fetus, it is called as Erythroblastosis Foetalis, and it leads to anemia. Hemolytic anemia, it is called. Hemolytic breakdown of RBC. As we know that RBCs, they goes to liver and spleen after their life cycle is completed. This can lead to elevated level of bilirubin formed due to hemoglobin breakdown, rise in bilirubin levels leading to jaundice. This is a fatal condition. The fetus may die. The fetus may die during pregnancy or just after the birth. Within 1 or 2 days, the fetus may die. Now, how to prevent it? Prevention. Prevention can be done by administering anti-RH antibodies to the mother just after delivery of the first child. Administration of anti-RH antibodies to mother when after delivery of first child. After delivery of first child, there are several recombinant antibodies are available in the market for this purpose, which are administered to the mother just after delivery of the first child in this type of cases to avoid erythroblastosis foetalis. So this is all for today's lecture. I hope I have made myself very clear to you. In case you have got any doubt, please feel free to comment. Please like and subscribe my channel, NCERT classes. Thank you.